Now let's talk about the red blood cells. So red blood cells or erythrocytes are biconcave disc shapes. So you can see here what they look like. They kind of look like a donut without the hole in the middle. It has a protein on the inside. That protein is known as globin, but because it's in the red blood cell, it is called hemoglobin. So you'll find that same protein in muscle tissue, for example, and we would call it myoglobin. So hemoglobin is the oxygen-carrying protein in your red blood cells. If it has oxygen attached to it, it is called oxyhemoglobin. When there is no oxygen attached, it is known as deoxyhemoglobin. Red blood cells do not have a nucleus, and they don't have mitochondria either. And because of that, the red blood cell cannot divide. Okay, so the red blood cell has a very short lifespan, 120 days, and then it dies off. It does not have the ability to divide. And because of the lack of mitochondria, it cannot produce ATP through Krebs cycle or the electron transport chain. It can only produce ATP through glycolysis. For the red blood cell count, which is the number of RBCs in a cubic millimeter, it is around 5 million per cubic millimeter. In an adult female, the range is from 4 million to 5.5 million. Adult males is a little bit higher, so from 5 to 6 million. And in children, it's a little bit less, so 4.5 to 5 million per cubic millimeter. And again, the lower numbers in females would be due to menstruation that occurs every month. Red blood cell counts are useful to diagnose diseases and to evaluate progress and response to treatment. So if you take an RBC count and it is low, okay, being anemic can either be due to maybe iron deficiency, or it could be secondary to another disease, meaning that there's something else going on in the body, and the body is responding to that by not making enough RBCs. So again, your RBC count could help it's kind of like a red flag that tells you that there is something going on in the body. And it's also a good way to evaluate the response to treatment and the progress of the patient. So if you give patient treatment and you find that the RBC counts are getting better, that means that person is responding well to treatment. Changes in RBC count reflects the changes in the blood's capacity of carrying oxygen. So in blood's oxygen carrying capacity. Now, how does the body control the production of RBCs? And that process is known as erythropoiesis, which is the formation of RBCs. We know that RBCs are formed in the red bone marrow, but what stimulates that process? So the story all starts if a person goes through hypoxia, so low blood oxygen levels, that will stimulate the kidney mainly and the liver to produce a hormone called erythropoietin. Erythropoietin through the bloodstream will reach the red bone marrow, and that stimulates the red bone marrow to increase its production of RBCs. Now that we have higher levels or higher numbers of RBCs, and that leads to the increase in the oxygen carrying capacity, by negative feedback, it'll inhibit the kidneys and liver from making more erythropoietin. What are the dietary factors that our bodies need in order to produce RBCs? Well, they're vitamin B12 and folic acid, and those are both required for DNA synthesis that is necessary for growth and division of all of our cells. And kind of a routine thing now that if a woman gets pregnant, she would be taking prenatal vitamins, and included in those prenatal vitamins would be vitamin B12 and folic acid to make sure that there's enough of them for all of that new DNA being produced and to prevent her from becoming B12 and folic acid deficient. We also need iron is required in order to produce and synthesize hemoglobin. What is the life cycle of a red blood cell? Well, it all starts with the nutrients that we take in. So remember, we need iron, vitamin B12, and folic acid. So when we eat those and those are absorbed through the small intestine, they are going to be transported to your red bone marrow. And in the bone marrow, the process of erythropoiesis will be stimulated with the production of these RBCs. RBCs will circulate in the bloodstream for about 120 days. And then when they are old, so at the end of those 120 days, they are going to be eaten up by macrophages in the liver. So macrophages in the liver are going to eat up and phagocytize these old RBCs. And then the hepatocytes, or the liver cells, will break down the older red blood cells and start the breakdown of hemoglobin. It'll break down into heme and globin. Well, globin is actually a protein, so the body is going to reuse that, recycle it, in order to make new hemoglobin. As for the heme part, that will break down into iron and biliverdin. Iron, again, will go back to your bone marrow and be recycled to make fresher and new RBCs, while biliverdin will change into bilirubin, and that will be secreted in the bile. And it is due to the greenish color of that. That's why bile is also green in color. And then they'll be secreted or excreted with feces in the small intestine, and in there they change into a product or a chemical known as stercobilin, and that gives the stools or the feces their very characteristic brownish color. Now, anemia, that is the decrease in the oxygen carrying capacity of blood due to the deficiency of either the red blood cells or the deficiency in the hemoglobin. Anemia is actually a misnomer, meaning that it is not a correct name. A means no, anemia means blood. So it's not that the person doesn't have any blood, it's just that the red blood cells are deficient, either in the cells itself or the protein carrying the oxygen. So the different types of anemia, we've got aplastic anemia caused by toxic chemicals or radiation, leading to the damaging of the red bone marrow. There's hemolytic anemia caused by toxic chemicals, where the red blood cells are being made, but they are being destroyed by these toxins. So hemolysis means the breaking down or the destruction of the red blood cells. Another type is iron deficiency anemia, and that is the most common kind of anemia due to the dietary lack of iron. So the red blood cells have deficient hemoglobin, leading to the decrease in the carrying capacity of these RBCs. Pernicious anemia, also known as megaloblastic anemia, because these cells are huge in size, and it's an autoimmune disease caused by the inability to absorb vitamin B12. So B12 is needed in order to make a healthy RBC, as we know. In this autoimmune disease, their body lacks the ability to absorb vitamin B12, leading to these huge cells, big in size, but they're very fragile. And that is known again as pernicious anemia. Pernicious in the fact that it is very hard to treat. Okay, and again, because we have these huge RBCs, they're known as megaloblastic anemia. Sickle cell and thalassemia are both genetic diseases. They occur due to defective genes. In sickle cell disease, under stressful conditions, whether that stress is due to low oxygen or due to maybe psychological stress, the RBCs change their shape into this sickle cell. Now, these sickle cells can get lodged into and block blood vessels, okay, leading to very painful conditions. Thalassemia is also known as the Mediterranean anemia because it is more common in individuals that come from areas surrounding the Mediterranean Sea. Again, both of these are due to defective genes, and in thalassemia there is a deficiency in the hemoglobin, and the red blood cells are very short-living.